Here we go. Okay, so I've talked about how we classify life. Now let's get the actors onto the stage. What I want to focus on this week is what are the basic types of living things that we have to deal with. Once we're there, then we'll be in a position to start talking about the things that they do. And the very highest grouping in Linnaeus's hierarchical classification is called the domain. And we can put every living thing that we know of into one of three domains. The one that you're in is the eukarya, or you'll also see it called the eukaryotes. And a eukaryotic cell, remember that all life is divided into these subunits called cells. Every cell has a flexible outer covering that lets some things in and keeps others out. That's called the cell membrane. And in a eukaryotic cell, inside the cell, there is a flexible sac called a nucleus. And the nucleus is where genetic material, the DNA, is stored. And we will talk in painful detail about what DNA does. Coming up about midway through the course is when we will hit that. Eukaryote cells are also divided into compartments. There are lots of specialized little compartments that carry out different functions. The compartments are called organelles, analogous in a way to the organs in your body, each one of which is specialized to do something different. You know, your heart, your liver, your kidneys, all those things, each one does something different that contributes to, well, how you live. So eukaryotes have nuclei and they have organelles. You are a eukaryote. You do have some cells in your body that don't contain nuclei, notably the red cells in your blood, but they originally had nuclei and they lose them in the process of being formed down in your bone marrow. So you're still a eukaryote, even though some of your cells lose their nuclei as they're mature. They originally came from cells with nuclei. Right. Cells that don't have nuclei and don't have those compartments, that don't have organelles, those are called prokaryotes. And there's two basic domains of prokaryotes, Archaea and Bacteria. Now, Archaea and Bacteria outwardly look very similar. The differences between the two involve a lot of fairly deep biochemistry. And this is going to be one of those times when I just kind of wave my arms and say, we're not going to get into that. That's not because you're incapable of understanding it. It's because we have a lot of material to cover in this class, and we have limited time. So in the interests of getting everywhere I want to go, this is one of those things that I'm just going to kind of wave my arms and pass through. We're not going to really talk about the chemical differences between Archaea and Bacteria. They are pretty deep and very important, but we're just going to say, okay, they're there, and we're going to keep on going. Many Archaea, not all of them, live in what we call extreme environments, environments that are so harsh that almost nothing else can. And organisms like that are said to be extremophiles. They can live in very acid or very alkaline or extremely hot or saline, salty or cold environments where almost nothing else can live. There are Archaeans that would live perfectly happily in an instant pot that was on and running, or even a pressure cooker. And not all Archaeans are extremophiles, but there's some famous ones that are. So let me show you what this looks like. In an upcoming lab, you'll see eukaryotic cells. You'll see this in the microscope lab. On the left are some cells from the inside of a human cheek. They've got a flattened, kind of slightly crumpled sheet of paper shaped to them. And in the center of each one, you can see a dark blue spot. That's the nucleus. In the microscope lab, you'll also see leaves of a water plant called Elodea, just a plant that grows floating in water, and it's got very thin leaves. On the right, that's a picture of an Elodea leaf under the microscope, and inside each cell, you can see a bunch of green circles. Those are organelles. Elodea cells have nuclei as well. I promise you they're there. You can't see them without adding special stains to bring them out. There's a lot of structures inside cells where you have to add a dye to make them easily visible because they're transparent under ordinary light. So your cells are eukaryotic. You are a eukaryote. Plants are eukaryotes. A lot of the complexity of a eukaryotic cell can't be seen with a light microscope. So we turn to what we call a transmission electron microscope. Here's a eukaryotic cell. Don't worry about what all of this stuff is called. What you need to see right now is, first of all, the very dark spot at the dead center of the cell, the thing that's black, is inside the nucleus. It's called the nucleolus, like a nucleus in a nucleus, and we'll talk much later about what that does. Surrounding it, you have this grainy zone. That's the actual nucleus. And around the nucleus, you can see there's some open spaces and some spaces that are filled with something and various grainy bits and bobs. We'll talk much later, about 3/4 of the way through, we'll finally get to what those bits and bobs are. And we'll go over the names of many of them. All I want you to get right now is the sense that this eukaryote cell is compartmentalized. It's divided up into many parts that do different things. A eukaryotic cell is kind of like a typical building on this campus. You know, my building, the Lewis Science Center, for example, has rooms that are used as offices. It's got rooms that are used as labs. It's got rooms for storage. It's got bathrooms. It's got rooms for maintenance, you know, where they put the heating and air conditioning equipment, things like that. Many different rooms for different functions. That's the way a eukaryote works. That's a bacterium viewed under the same microscope. And what I want you to see is that it's not compartmentalized. There's no nucleus. It's kind of grainy and light colored in the middle, but that's not set apart from the rest of the cell. Basically, all of a bacterium's metabolism will typically take place in the same wide open space. This is kind of like the football practice facility, which is basically a roof over one very big room, half the size of a football field, where the football team can practice when the weather's no good. So the football practice facility is much more like a prokaryote because it's all one big room and everything happens in the same space. So that's the difference between a eukaryote, which it looks like this, and a prokaryote. This bacterium is a prokaryote, and Archaea are also prokaryotes because they've also got that open plan with no nucleus and with no division of the cell into compartments. Mostly not. I can think of some exceptions, but for what we need to do here, this will work. Seen with a transmission electron microscope, you have bacteria on the bottom. Those are archaea on the top. They're both very small. We measure small things in microns, and a micron is 1/1,000,000th of a meter. It's also called a micrometer, but we often just say micron. Let me see, for those who don't know, if you're not familiar with the metric system, a meter is roughly 3 feet and a few inches. A thousandth of a meter, a millimeter, is about as thick as the wire that makes up a largish paper clip. And a micron is 1/1000th of that. So you could fit 1,000 typical bacteria in a line that was as long as the wire on a big paper clip is thick. If that helps you visualize the size that we're talking about. So as cells go, this is very small. Eukaryotes on average are bigger, although there is some overlap. And the differences between archaea and bacteria involve some fairly complex biochemistry, which we will not cover, not because you can't understand it, but because we've got a lot more material to go. This is why the gods gave us Wikipedia. You can look this up if you need to know about ether-linked membrane phospholipids instead of ester-linked phospholipids. The only thing I'm really going to say about archaea is that the most famous ones live in extreme environments. Somebody's mic is on and making squeaking noises. If you wouldn't mind muting whoever it is. Now, somebody's mic was making little squeaks, so if you could mute it, that would be great. This is an aerial photo of Grand Prismatic Spring in Yellowstone National Park. For scale, down at the lower right, you can see there's a walkway with people on it. So that tells you how big this is. This is a hot spring, and it's not one you can bathe in. There have been cases of people jumping off that walkway and jumping into the spring and dying horribly. The water temperature is pretty close to boiling. If you jump in here, you will get severe burns in a few seconds. So you can't live in here. You can't swim in here. Okay, someone please mute your mic. I'm getting a lot of squeaks and squawks I'm hearing. Brennan, mute your mic. Okay, Brennan, please mute your mic. Thank you.
I think you may be able to do it from your side.
Okay, well, I might have to stop the share to do it. It's easier if I just yell at him. Hang on, let me see if I can. Is that him? Okay, I think I just did it. Yeah, I think I just muted him. Maybe I didn't. Hang on. Okay, I think I did it. Okay, great. Right. All those colors, especially the oranges and the greens that you see in the Grand Prismatic Spring, are caused by mats of archaea living in these huge masses in water that is uncomfortably close to boiling. You can't live there. They do just fine. That's one of the things that archaeans are capable of doing. That's a river in southern Spain, rich in heavy metals, which is why I like it, and is about as acidic as lemon juice. On the pH scale of acidity, if you're familiar with that, it is about pH 2. There are archaeans living perfectly happily in this river. Nothing else can. No fish are going to make it. It's loaded with lead and cadmium, and it's very acid. But there are Archaeans that are adapted to live here perfectly well in the Rio Tinto. Probably the most extreme is this is deep under the ocean, maybe about 5,000 feet down, probably more than that. It's called a black smoker because what's happening is that superheated water is coming up out of the ocean floor like a hot spring, except this is coming out at a temperature that's greater than the temperature of boiling water on land. And it looks black because it's loaded with iron and sulfur. So it looks like smoke. It's actually this chemical-rich, superheated water. And there are Archaeans that live perfectly happily in the outflow of one of these black smokers that can tolerate temperatures as high as 220 Fahrenheit or 104.5 Celsius, if you prefer that. So there are Archaeans that are extremely tough. And that's the main thing I want you to know about Archaeans, is that many famous ones are famous for being extremophiles. Totally extreme, man. Right. Okay. Now to move on to bacteria. Most bacteria are at most a few microns in size, and even under very good light microscopes, they look like this. Each one of these little purple dots is a single bacterial cell. And you can tell there's not a great deal of detail that we can make out using light microscopes. Most bacteria, not all, are translucent when they live. If any of you go on to do any, take microbiology, or even if you get into, medical laboratory science or something like that, if you work as a lab tech, like I used to a long time ago, you'll learn how to do various chemical processes to add stain to bacteria and make it possible to see them. These bacteria have been chemically stained. They're not naturally purple. How you classify bacteria is a complex business that we don't need to get into too much of, but one of the criteria can be the shapes of the cells. These bacteria are called cocci, and they're shaped like little dots. So you can see they're basically round. Some of them are kind of paired up, but these are cocci, dot-shaped bacteria. Rod-shaped bacteria, shaped like rods. If you could see one up close in 3 dimensions, it would look like a sausage. These are bacilli. Bacilli is Latin for little rod. And up there at the top left, you can see a couple of bacilli up close. Someone once said, "Hey, do you know the shapes of bacteria?" And I said, "Don't be silly." Just keeping everybody on their toes. Right. Spiral-shaped bacteria are called spirilla. Just one is a spirillum. So there's a bunch of spirilla up at the top. And there are bacteria that are, you could think of these as extremely short spirals. They're just kind of shaped like a little curve, like a comma or like a bean. And we call those vibrios. And down at the bottom, you've got a transmission electron micrograph picture of a vibrio. On the other hand, very long spiral bacteria are called spirochetes. Chete is Greek for a bristle. So spirochete means spiral bristle. And here's a couple of spirochetes right there. The bacterium that causes syphilis is a spirochete, incidentally. So is the bacterium that causes Lyme disease, which is what you can catch from getting bitten by a tick. One of these things you can get around here in the summertime because, as you know, if you've set foot outside in Arkansas for more than 5 minutes in the summer, we've got a lot of ticks. And the bacterium that ticks past to you that causes Lyme disease, that's a spirochete. You can further identify bacteria by how they group together. These are cocci, and they're growing in bunches. The Greek word staphylos happens to mean a bunch of grapes, so we call these staphylococci. Again, I will never test you on whether you know what ancient Greek words mean. I'll never ask, what's the Greek for bunch of grapes? But some people do find that knowing the meanings of the words helps them learn the vocabulary. So if that's true for you, then this is what it means and you can know it. If it's not true for you, don't worry about trying to memorize staphylos. But these are staphylococci growing in bunches. If anybody's ever had a staph infection of the skin, staph is short for staphylococci. Bacteria like this can cause skin infections. If you've ever heard of MRSA, MRSA just stands for multi-drug resistant Staphylococcus aureus, that being a species of Staphylococcus that causes skin infections. So that's where you might have encountered the word staphylos. I used to see these a lot when I was a medical lab tech at the Berkeley Free Clinic in California, because bacteria that look like this cause gonorrhea. We did a lot of sexually transmitted disease screening. Probably don't really want to hear too much about that, but it is something I have some experience with. Bacteria that grow in, or cocci that grow in pairs, or sometimes in this picture you can see little quads of them, groups of 4 like a clover leaf. If they grow in pairs, they're called diplococci. Diplo happens to be Greek for double. And gonorrhea and also bacterial meningitis happen to be caused by diplococci. And then bacteria that grow in chains, the Greek word for twisted like a rope is streptos, and these are some streptococci. If you've ever heard of or had strep throat is caused by a particular type of streptococci. So we use these a lot, especially in medical settings when we're trying to identify what sort of bacterium is causing somebody's symptoms. I used to do that quite a lot for several years. Some of you might encounter these at some point in the future yourselves. To do better than light microscopes, we have to use electron microscopes. But even there, you can see there's not a great deal of detail on the inside. So that's a slice through a bacillus taken with a transmission electron microscope. UCA does not have a transmission electron microscope, by the way. UAMS does, but we don't. UCA has a different type of microscope called a scanning electron microscope, but not something that can take pictures like this. So in this slice through a bacillus, you see it just kind of grainy inside. There's not much detail. It's kind of, grainy gray fuzz. That grainy gray fuzz is surrounded by a flexible covering called the cell membrane. It's abbreviated CM here. There's a few bacteria where that's all there is, but most bacteria have a stiffer outer covering called a cell wall. So the membrane is flexible, but the membrane is pushed up against the cell wall, which is a lot more rigid and gives the bacterium its fixed shape. The reason why bacilli have this rigid rod shape to them is because they're surrounded by a rigid cell wall. Imagine taking a filled up balloon and stuffing it into a cardboard box. You'd have a decent model of a bacterial cell, with the balloon being the cell membrane and the firmer cardboard making up the cell wall. There's another one. Again, you've got grainy stuff on the inside. You can see the cell wall on the outside. What it's made of is different in different types of bacteria. That's important clinically, but we don't need to get into it here. Let's just say that at least some of the wall, and in some cases almost all of it, is made of a compound of proteins stuck together with sugars that makes a very rigid substance called peptidoglycan. By the way, if I italicize a word on a slide, that often means it's something I would expect you to recognize on a test. So if you want to make flashcards or notes of vocabulary that I would expect you to know, a good start would be to go through the slides and look for anything I've put into italics. The reason peptidoglycan matters is that antibiotics of the penicillin family, which includes not just regular penicillin, but things like amoxicillin and ampicillin, cephalosporins, things you might have been prescribed. I mean, my kid used to get amoxicillin for ear infections. Some of you might have taken these before, or you might know them. The reason they work is that they block bacteria from making peptidoglycan. Now, that makes them very safe for humans to use. A lot of things will kill bacteria, but they would kill your cells as well. I mean, a point blank shot from a 45 will kill bacteria, but it'll also kill you, or certainly inconvenience you a great deal. So when you're coming up with drugs to kill bacteria, you want something that'll kill the bacteria but not harm human cells. And penicillin and its kin fit the bill very nicely, because what they do is interfere with the bacterium's ability to make peptidoglycan, which means that a cell can't grow if it can't make more cell wall and expand. So that's why penicillin was called a wonder drug when it was introduced first in World War II for soldiers, and soon after World War II for the civilian population. That's why it was such a game changer. It would kill bacteria, many bacteria, not every one, and leave your cells standing. And it saved countless lives because it can target peptidoglycan and nothing that your own cells need to live. This may seem like trivia, but we're actually going to come back to this about 2/3 of the way through. Outside the cell wall, there may or may not be a layer of snot. That's called the capsule. These bacteria have been stained with ink, and you can see there's a unstained, completely light halo around the bacteria. That halo is where the snot is. That's called the capsule. This can be important clinically in determining whether a bacterium causes infection or not. And I'm bringing this up because we are going to come back to bacterial capsules in a later lecture. They turn out to play, they turn out to score an assist, to play an important part in a much later biological discovery that's very important. So that's why everything I'm saying about bacteria, either we will come back to, or it has practical implications in a field like medicine. Even if you're not going into health sciences, some of you might get sick sometime, and it may help you understand the treatment that you're getting, for example. So we are going to come back to this snot layer that some bacteria have and others don't, the capsule. I won't say too much about bacterial reproduction. They're capable of engaging in a rather, by our standards, strange type of sex. It's called bacterial conjugation. But typically, reproduction is just by splitting in two. This is a bacterium that was caught in the act. You can see it's a coccus, but there's a little furrow in the center. You're seeing a slice through it, by the way. And that little furrow is where the cell is pinching in two. There's two clumps of DNA, that light colored stuff. One is going to go to each daughter cell. So this is typically how bacterium reproduce. They split. There's a type of sex they can have that I have to talk about DNA a little more to be able to talk about how bacteria have sex. We may be able to get to that later. The very last thing I'll say is that some bacteria can form resistant capsules when life gets rough, and we call these endospores. These are pictures taken under the light microscope of two different types of bacteria, and the cells look dark, but you can see they've got these bright areas inside. The bacteria on the right have these bright areas inside. On the left, those bacteria have bright areas, but only at one end, so they kind of look like tennis rackets under the microscope. Those bright areas are called endospores. This is like the escape capsules, right? Like the one that C-3PO and R2-D2 used to escape the Tantive 4 in the opening of Star Wars Episode 4, A New Hope, which I'm sure you've all seen. When life gets rough, some bacteria can kind of package their DNA and some other things they need into this very thick-walled capsule that they make. And the capsules can withstand almost anything, sometimes for years, in a kind of suspended animation. They can just sit there, they do nothing, they're not obviously alive, but they'll come back to life when conditions improve. Remember I said it's sometimes hard to come up with a strict border between living and non-living? This is a case. A bacterial endospore doesn't do any metabolism or anything like that, maybe for years. So it's like it's dead, except it comes back to life whenever conditions come right again. So there's a little philosophical conundrum for you. And I mentioned this because the endospores of the anthrax bacteria can be prepared as a biological weapon. Officially, we're no longer doing this. If you inhale these endospores, you get severe lung infection that's usually fatal. I guess most of you were born after September 11th, 2001, but I'm sure you've heard of it. It's the kind of thing that, you know, if you were alive at that time, you remember exactly where you were when you saw the airplanes fly into the World Trade Center in New York City. In the aftermath of that, there were envelopes containing prepared endospores, which just look like a very fine white powder. Somebody, probably a disgruntled researcher at a US Department of Defense lab, although I'm not sure if he ever confessed, mailed those out to politicians and media outlets. 22 people got sick, 5 of them died. So endospores have been used as biological weapons like that, although I think officially we are no longer doing this. This matters because all of these features can be used to identify disease-causing agents, or as we call them, pathogens. A pathogen is a living thing that causes disease, and we can identify these in human patients, in animals and plants, in food, in water. If you remember times when some produce has had to be removed from grocery store shelves out of fear it's contaminated with something called E. coli, That's a pathogen. That's a bacterium that causes disease. So everything from food poisoning to strep throat to syphilis and gonorrhea to cholera is caused by bacteria. There aren't any archaeans that cause disease, certainly not that I've ever heard of. Viruses, by the way, can also be pathogens. You'll notice I haven't mentioned them. Viruses aren't made of cells, and they're another one of these cases that's on the in the gray zone between living and non-living. We'll talk about those later, but we can't really talk about them now until I've given you some background. It's often not the bacteria themselves that cause illness, but many bacteria release toxic compounds, toxins, that are released into the body, and they might affect the body a long way from where the bacteria themselves are growing. So tetanus and botulism, the most severe type of food poisoning, staph infections, and so on, the symptoms are caused by exotoxins that are being released by the bacterial cells and spreading through the body, maybe a long distance from where the actual bacteria are growing. There are also components of bacterial cell walls that act like toxins themselves, and we call those endotoxins. Endo means inside, the toxins are in the living bacteria. Exo means out, the toxins are spread, possibly a long distance from where the bacteria themselves are growing. Okay, if that wasn't clear, endo means in, exo means out, and exotoxins are released by the bacterial cells and spread through the body. That's a painting of a British soldier from 1821 dying of the disease tetanus. The actual bacteria were probably growing in a wound. They can get into the body just through a scratch. And the bacteria themselves may not spread very far from the wound that they get into, but they produce exotoxin that, in this unfortunate man's case, is circulating through his entire body. And the effect is that muscles can't relax. This is why tetanus also used to be called lockjaw, because the first muscles to be affected are the muscles that move your jaw. You can't open your mouth because your jaw muscles lock up. And in this case, every muscle in this man's body has locked up. That's why he's in this back bend, and his fists are clenched, and his toes are curled, and his arms are drawn up like this, and his whole back is bent. That's the effects of tetanus exotoxin. Its effect is to allow your muscles to contract but not relax, so you seize up like this. There's a related bacterium that produces another exotoxin that paralyzes muscles. This one can be deadly, but it's actually harvested, and the cats are fighting. Right. It's sold under the name Botox, and it's used when injected into facial muscles. It temporarily paralyzes the muscles, and it's used cosmetically to tighten your facial muscles and temporarily remove wrinkles. Makes your skin very smooth, but also kind of mask-like. Anyway, real quick, bacteria cause disease, but the overwhelming majority of them are harmless. They're everywhere. Water, air, soils, your body. You've got more bacteria in your intestine than there are humans on the planet. Every one of you does. And most of those are quite beneficial. You probably didn't want to know this, but by weight, about half of every bowel movement is bacteria. Now you know. You can't unknow that. There are bacteria that require oxygen, and we call those aerobic. But many bacteria are actually poisoned by oxygen and they have to live in oxygen-free zones. We call those anaerobic. Many bacteria are involved in decay. We call those decomposers. We'll get back to those at the very end of the course. And there are bacteria used in the food industry. They convert milk to buttermilk and yogurt. If you're eating yogurt, if you've ever eaten yogurt, you've eaten live bacteria. They break down waste in sewage. I know I'm going a little over, leave if you have to, but I'm almost done. There are bacteria that will break down petroleum and clean up oil spills. Bacteria put the holes in Swiss cheese. Bacteria growing on the roots of soybeans and other legumes make the soil fertile and make the plant grow by converting nitrogen into a usable form. And a healthy human body contains thousands of species of bacteria that are critical for human health. And if you've ever taken a probiotic supplement like this, you're taking helpful bacteria into your body that supposedly can improve your health and replenish your good bacteria and get rid of the bad ones. So I went a tiny bit long, but by the power vested in me by the state of Arkansas, I will now